Yes, welcome everybody to yet another Friday afternoon webinar with NORA. In this week's NORA webinar, you will meet Andreas Kleppe from the Institute for Cancer Genetics and Informatics at University Hospital. Andreas is a researcher at the Norwegian Research Council's ICT Lighthouse project called DoMore, where the goal is to use medical computer vision to develop automatic systems that could improve the treatment of cancer patients. He also holds an associate professor position at the Department of Informatics at the University of Oslo. His research interests include machine learning and image analysis, in particular medical computer vision. In several of the projects he has been involved in, the goal has been to develop automatic systems that use various image data to predict whether a patient will recur and die from a cancer or survive following surgery. So here, Andreas, the algorithm really could be the difference of dead or alive, actually. The title of the talk is Evaluation and Generalizability of Deep Learning Systems. So please, Andreas, the screen is yours.
Thank you. It's nice to be here. Thank you for the invitation. So yeah, I'm going to talk about the evaluation and generalizability of deep learning systems. Just a slight disclaimer here, views are my own. That doesn't mean that I don't share the views of many of my co-authors, but just so that I can speak a bit freely also about subjects we haven't discussed. But we mostly agree on everything, I think. So, what is supervised machine learning, going all the way back to basics? In image classification, we usually have an image and we want to predict the probability of a certain set of output classes, either the individual probabilities or just the final classification. We have done this many years before my time also. And the classical approach was that we applied some handcrafted features, feature extraction step that was computed for the image, and then applied a set of training images to learn associations between these feature values and the classes, the true classes, to get some prediction of the probability of each class. That might have worked okay for this simple classification tasks, but as the tasks grew more complex, this was not always sufficient. So one started to create multiple feature vectors. Multiple features. So creating a large feature vector and applying then a more sophisticated classification methods to get the predicted probability. So one started to leaving more and more up to the data and to the machine learning, because it was this step here, the classification method, that was the machine learning step. The feature extraction was traditionally not machine learning, it was handcrafted features. But as time progressed, also this step started to becoming data-driven. We had this bag of visual words, for instance, which created this code words, localized image features from the data itself. So it became also more data-driven in this part of the classical machine learning setup. But in 2012, the winner of the ImageNet competition was a deep neural network. And from that time and onward, it has been, the machine learning has been primarily focused around these deep convolutional neural networks for image classification. So this process is basically completely data-driven. Of course, there are many design choices here, but it's directly from the data to the target output through these deep networks. The main the shift here towards relying more and more on the data means that we need to be means that the model will adapt more and more to the training data it has seen, and that makes it more and more important to evaluate how the performance of the trained model is on new samples. So already in the classical machine learning, we had this training and test setup where we had our development data set, and we typically split it into random subsets. So we had this training subset, or that could also be a training and tuning subset, as we call it. Some call it training and validation subsets, where we train our model and adapt our hyperparameters. And then we had our test subset, where where the performance is being evaluated. And it was it has been known long in the machine learning community that the test set should be kind of kept in a vault and only brought out at the end of the data analysis. If we don't do this, if we repeatedly evaluate the test subset and choose the model with the smallest error, then this model and the error estimated on the test subset might be underestimated, sometimes substantially. This is kind of basic machine learning knowledge from way back. Also, Russell and Norwig, another common test book, writes similarly. If you don't like the result on the test subset, you have to obtain and lock away a completely new test set if you want to go back and find a better hypothesis. So we shouldn't be repeating the analysis on the test subset. So that's the setup one usually has, or at least had. But an important question is that how representative is the development data set of the entire intended application? The intended application is typically quite broad. Might be some particular subjects, some particular image devices, and so on, but it's not usually not restricted to for instance, particular parts of the population, or sorry, I shouldn't be looking at the chat. It's probably not restricted to a particular country, for instance, or a particular racial background, as some of this has been shown to actually be in practice. So it's a question: how representative is the development cohort of the practical application you intend? So usually we kind of had assumed that the development data set was realizations of some independent and identically distributed random variables, but that is not usually the case. We can see a couple of examples here to indicate whether a test subset is sufficient or not to evaluate the performance of a system. For instance, here, Wickler and colleagues analyzed a deep learning model, which used these thermoscopic images to predict the probability of melanoma. They showed, using some explainability technique, that the model was influenced by skin markings, which had been placed beside the object of interest in the image. Obviously, this is something that was present in the training data, and therefore the data-driven deep learning approach was able to associate these skin markings with the probability of melanoma. But in practice, we would now want to rely on the skin markings to make our predictions, we would of course make the predictions based on the object itself. So kind of it had learned some spurious feature that did correlate with the outcome, but it was not the intended features. And in a practical settings where you might only have the object of interest, the system might perform less well, or maybe even quite poorly. So the test subset performance would then not have been representative of what the system would perform in practice. Another example by Sek and colleagues used also deep learning and investigated a model that detected pneumonia in chest radiographs. So here they had multiple development data sets from multiple institutions, and the institutions had different disease prevalence. And it turned out that the radiology technicians placed these metal tokens on the patients before imaging. And the deep learning network were able to utilize these distinct features between the institutions and combine it with the difference in disease prevalence to make better predictions. Of course, it's not the metal tokens that determines whether or not the patient has pneumonia. So again, it's these spurious features that might be learned by deep learning networks and might be represented in both training and test subsets, but would not in general be there for new data. So random test subsets are similar to the training data, and this might cause the performance to be overestimated because of these spurious data features that correlate with the target outcome only in the development data. We've seen a couple of examples here that could be explained by explainability techniques, but in general, it would not always be possible to find these spurious features. So even though in some examples it is possible to use explainability, it will not always be like that. It might also be that the important predictive features are not adequately represented in the development data. And if they are not in the development data, they're not in the test subset either. But they would be, however, more likely at least in an external data set. So we need data external to the development data to obtain more realistic performance estimates. Going back to our sketch here, we have that we have an intended application of our systems, the model we develop, and we want to not only have the development data set, but we want to have also a separate independent or external data set that is not just a subset of the development data set, but that is a new data set, for instance, from a different country or a different institution than used for development. Ideally, the external dataset should be representative of the intended application, and if so, it would provide an unbiased performance estimation of that application. Of course, in practice, it would not be representative, just as it was difficult to obtain development datasets that is truly representative of the applications. The external dataset will also not probably be representative of the entire intended application. But if we do get good performance in the external dataset, that indicates firstly that we have good generalizability of one of multiple intended settings. And secondly, if there is no relation between the development we did, the development dataset and the selected model, and the external data set, then this suggests that the model will also generalize to new settings, because if it works well here, it's quite unlikely that that was the only other setting it would work fine on. It will probably be many. In the medical statistical community, there is this type categorization of type of prediction model studies that uses simpler approaches for machine learning than deep learning, but they have this division into studies based on how reliable it is and what type of data it uses. And the main distinction is that you have kind of this one data set studies versus this validation on external data set studies. I should mention, I was perhaps not entirely clear, but the validation here is then separate from the development data in the machine learning community. Of course, many use validation as the data set to, or the subset to tune hyperparameters. That is not what the term is here. We use tuning for that hyperparameter data set. Validation is the separate final test subset, test set, not subset. Okay, so but how to use these external data sets then? Just as it was for the test subset, the performance estimated using an external data set become over-optimistic if multiple prediction systems are evaluated on that external data set and the best one is selected. So the external data set should ideally be used only once to evaluate the final prediction systems. And ideally, also the details related to this evaluation should be pre-specified. For instance, which subject it is, which data it is, and even which performance metric you use. If you do any post-hoc adjustments, evaluate the external dataset multiple times, it might bias the performance estimation, just as the case was for these test subsets. We recently published a perspective in Nature Reviews Cancer, where we advocate such pre-specification before evaluation of an external cohort. We had this, we proposed this set of protocol items for writing up a protocol or a document before you do the evaluation to just specify each part of it, the model, and what type of analysis you plan to do. And you should then, we argue, select one primary analysis, which is kind of your main. Sounds a bit rigorous, and it is. One positive side of it is that you, if you are a large team, you would be more forced to discuss between yourself and what to do and precisely what to analyze. And you would, of course, then not have the opportunity to adapt to the data, to the external data set. We reviewed some of the recent literature in deep learning on cancer diagnostics. It was quite a lot, so we restricted ourselves to what we assumed was influential studies, and these were the ones with at least 20 citations per year or published in a journal with impact factor 10 or larger. What we saw was that the external cohort evaluation is becoming quite common in such influential studies. We see here that there is a quite steady and steep trend to increase performance, increased proportion of studies that evaluate an external cohort in these influential studies. So it's more than 2/3 that now evaluate external cohorts. And it was particularly those published in journals with high impact factor. They tend to require that now. So that's probably the reason. However, we saw that the external cohort evaluation was rarely reported to be predefined. So among the 50 studies we found, among these presumably influential studies, and it was 50 studies that evaluated external cohorts, but only 8 of them had reported to predefine the primary analysis, which is quite low. Some others might have had a predefined primary analysis, but at least it wasn't reported in their paper. So we don't know whether they had done this post hoc adjustment after looking at the external cohort the first time. And in some studies, it was quite apparent that they did evaluate the external cohort multiple times, and then you wouldn't know whether it's over-optimistic or not. So we propose these possible steps in the development and evaluation of deep learning systems, where we start out with this basic studies, where you have, where you have an idea and you want to just check it out, whether it's something you want to invest your time and resources on. Maybe you have just a sparse amount of data and you make some simple pilot and see, is this something to move forward with? And then you have this large set of level 2 studies where you often try to obtain as much development data as you could. You test a lot of different hyperparameters and networks and approaches and then you end up with a system you think this is going to work in general. This is, of course, here that many work, and important contributions are here. But to get into practical environments, we should evaluate new cohorts. We separated this level 3 from level 4 studies, where in the level 3 study, you have an external cohort evaluation, but you do these post hoc adjustments, and you then possibly end up with biased performance estimates. While in the level 4 study, you have this pre-specified primary analysis, and you do that, and that is kind of your main result, and that is an unbiased performance estimation of the setting represented by the cohort. And we argue that the successful level 4 studies might warrant that one should start out with these clinical trials, and that you could actually jump all the way to phase 3 trial. Now in the corona times, you probably heard a lot about clinical trials. Phase 3, then you have gone a bit ahead. You have done with safety, and you're done with some dosing and stuff, and you are kind of in the setting where you want to test this out in practice, in medical practice, and see the utility. We argue that even with retrospective data, you can come quite a lot in estimating the performance of your system. So then the question becomes, okay, so we are here in the level 2 study, and we wanna develop a system that with high degree of probability will work in new cohorts. Because if we are to be this rigorous and just evaluate the external cohort once, we must be quite certain before we proceed with that evaluation that the system will actually work. Because if not, we have to acquire a new cohort. So then we are in the second topic I want to discuss today, and that's to facilitate generalization. There is a lot of possibilities here, and coming from classical machine learning, what one might think that the best approach is to reduce the number of adjustable parameters, the weights in your deep learning network. Of course, that's possible, reducing the number of layers, nodes, maybe applying other techniques such as depth-wise separable convolutions. But in general, that might not be the best approach, but it could be tested out. Another possibility is to control the network's capacity, so its ability to express complicated relations between input data and target output. You could do some regularization, you could do dropout, weight sharing, many techniques to control the capacity. Another popular approach is, of course, transfer learning. So if you have limited data, you might not want to learn the entire set of weights, but you instead start out with the weights of some model trained on ImageNet or something else. All these approaches, these three first, might increase the generalization. But in particular, it might increase the generalization to test subsets because it doesn't change the input data. So it kind of makes it more robust, you could say, and it should then generalize better beyond the training subset. But whether it will also generalize beyond the development data, so the combination of the train, tune, and test subsets, that is more uncertain. For that, you might want to consider, in addition, maybe, these other three items here. For instance, data normalization, applying some algorithm before training and before evaluation that makes the input data more similar. You might do data augmentation, altering the input data before inputting to the network during training to allow the network to see a larger variety of input data. And you may, if possible, also employ more diverse training data. So I want to exemplify this by my field of research in the recent years, which is where we have applied the deep learning to histopathology section images. So these are standard images that the pathologists look at in routine clinical practice. For instance, they could be obtained by you having a patient and you resecting the tumor out, and then you divide this specimen up into a series of tissue blocks. You take a thin section from the tissue block, for instance, 3 micrometers, and you place it on a glass slide. You apply this standard staining called hematoxylin and eosin staining, and you place another glass on top, cover slip it, and then you scan it on a high resolution scanner. The pathologist usually looks at this in a microscope, this glass slide, but we will just scan it and then obtain these sort of images. The resolution here is quite high. Each pixel has about a quarter microns per pixel. So it's similar to what the pathologist will look at in a microscope with approximately 400 times magnification, which is commonly used. And the dimensions here are approximately 100,000 pixels in each dimension. The pathologists do a lot of assessments using these images. For instance, they detect cancer here. What they do not do directly is that they don't directly predict the outcome of the patients. How will it turn out with this patient? Will it relapse, recur the cancer a few years in a few years, or will it survive and be cancer-free for many years? We tried to use deep learning to use these same images and then train a network that will directly predict whether or not the patient will recur and die of the cancer after surgery. And we published this in the Lancet last year. So we had this setup, which I already sketched. In training and tuning, we applied 4 cohorts with approximately 2,500 patients. We had this test subset, which was new samples, but not really new patients that we just used to be a bit more certain before we did this validation. And the validation was done, of course, pre-specified, so that even the primary analysis was pre-specified, so that we knew on forehand precisely what to evaluate, which model, which metric, and so on. the approach we applied was that we first took this this input image, we downsampled it and fed it into a segmentation network, deep lab segmentation network, and we got out this tumor delineation. That was quite accurate detection of tumor. So with this automatic tumor detection, we created these image patches within the tumor detection. We call them tiles, and extracted that from the image. We did this tiling at 2 resolutions, one at the full resolution called 40x, and one at the 4 times reduced, so that we get a more overview of the architecture, tissue architecture. We trained 5 models in each case and created an ensemble model, and then thresholded that ensemble score, and then obtained a classification. If both ensemble categorizations was poor, then we had one group. If both were good prognosis, meaning that the patient would survive long, then it was one classification, and if they disagreed, that was the kind of the uncertain group. So looking at the results in the validation, we obtained this group of patients that we predicted had a good prognosis. So they survived well. Within 5 years, their survival was more than 90%. So they had good prognosis in this cancer. The patients we predicted as poor prognosis, they had markedly different outcome. So they died more frequently of their cancer. Within 5 years, their survival was less than 70%. So the hazard ratio, the ratio between, it could be viewed as a sort of a relative risk, although it's adapted to survival analysis as this one. It was about 4, so it was about 4 times more probable of recurrence and death of cancer in this high risk group than in this low risk group. And we had this middle group. Notably, this middle group was quite small, less than 50% of the patients. So mostly the 2 ensemble classifiers agreed on the predicted score. Importantly, in applications, you need to also take into account what other type of markers are available, and in colorectal cancer, as this was, it's primarily this nodal stage that is giving prognostic information, but slightly also other markers. The important thing is that when adjusting for the prognostic effect of the other markers, our new marker was still markedly identifying which patients were having a good and poor prognosis, so that it could be used in addition to this clinically and pathologically available markers. For instance, the nodal stage is assessment of the lymph nodes, which we, of course, we didn't have it as input to our network, but it's resected along with the tumor, and then you see, then the pathologist see, is it tumor in the lymph node? Yes, no, and how many? Okay, so that's our Lancet results, and one important part there is, of course, that we had a lot of data. We had nearly 2,500 patients. We had 2 scanners as well, so we had about 5,000 images, each quite big, as you saw, 100,000 pixels in each dimensions before segmentation. So we had quite a lot of data. Millions of these tiles were inputted, and therefore we also had introduced our network for quite a lot of image variation. Here is just for quite random examples where you see that there is substantial variation in at least the color, and probably also more detailed differences between the images. Since we had that many training data, we probably had learned the network to handle this quite well. If we look at it on a tile level, it might appear even more distinct. So the network kind of had to learn to ignore much, because it was a large variation in the training data. But what if we then didn't have as much training data? What would happen there? So in this perspective article, we looked into this this setting, and also then how to mitigate it, because it's not always you have this large amount of training data. So here it's the training with the old cohort applied on evaluated on the external cohort, but trained on the four development cohorts, the 2,500 patients approximately. We reduced it to the size of the largest training cohort, but we did randomly sample from all 4 cohorts. So here it's about 1,000 patients included, and then 500 and 250 and so on. We see that the performance T-index is a measure of discrimination, and it quite rapidly decreases if we don't use as much training data. But could we compensate for this by artificially increasing the variation in the training data? So this was our original amount of color distortion and augmentation technique, which was quite modest. We didn't seem to need more than this, but we now investigated what if we doubled it, quadrupled it, and so on. Would that compensate for some of the performance loss that we saw when training on less data? And certainly it did. So here is training with the 1,000 patients again, but the result we got initially with the initial amount of color distortion could be markedly improved by increasing the amount of color distortion. And actually, with this 4 times amount of color distortion, the result on the external cohort was nearly as good as with all 2,500 patients, just by using these 1,000 patients and a lot of artificial color distortion during training. So that's certainly could mitigate for much of the performance loss. We also looked into what if we only had 1 cohort for training and tuning. I said that we'd selected 1,000 patients that matched the largest cohort we had, largest data set we had in training. We had 4, but it was a random sample across all 4. What if we only used the largest cohort? So it's still this same, it's still about 1,000 patients, but now we sample it just from the 1 cohort. And we see that even with a large amount of color distortion, it was not able to perform as good as with the matched random subset. So the subset across all 4 training, all 4 development cohorts initially used. But here and here, it's the same amount of patients. So it's important with both natural variation and artificial variation when training deep learning systems that should generalize well beyond the development data. And these 2 variations of data complement each other. Lastly, I just want to mention a couple of other studies, recent studies that have looked at something similar. They're still in the histopathology domain, but here it's Stake and College, which has looked into tumor detection, and you had this source domain, the development data domain, the target domain, which is the external dataset domain, and they looked into, okay, what if we do color augmentation as we did? That would increase the diversity, the variation, before presenting to the network during training. What if we did stain normalization, so make the input data more similar before inputting to the network during training and during inference. And you had this generative adversarial networks as well as tested to align the domains. I haven't talked too much about this today, domain adaptation, but that's also a technique that could be used. The downside is, of course, that you need samples from the domain you want to target. So yeah, we focus on this part. They applied quite extensive color distortion as we did. You see here that here is the original images from 5 centers. Each row is the 3 first rows is one type of scanner. The 2 last is 2 different. And they either applied then much color distortion. They applied stain normalization, not both, but just stain normalization, or this domain adaptation with cycle GAN. They saw that in one of the settings, in one of the datasets they investigated, all 3 methods worked quite well. While not doing anything caused a substantial drop from the development setting to the external cohorts, the external datasets. The 3 techniques all reduced the difference between development and target domain. While in the other setting they investigated, still tumor detection though, they found that both color distortion and stain normalization worked very well, while cycle GAN did not was not able to compensate much for the performance drop. One interesting part with this that you can do stain normalization instead of color augmentation is that the augmentation is kind of indicates that it's the size of the training data that is important, but stain normalization doesn't decrease the size at all, and it might suggest that actually the main point with color distortion or color augmentation is that it increases the variability of the input, not the size. They also looked at the activations and what maximizes the activations of the last convolutional layer, and then here are different filters, maximum activations. There's without any augmentation, normalization, or cycle GAN, and with augmentation, with normalization, and with cycle GAN. So interestingly, you see that when they did this color augmentation before training, the network learned to not use the color information. It should be noted that they did a very extensive color augmentation, so with less color augmentations, this might have looked differently. And it's also interesting to see that these cellular structures are appearing. Another group evaluated something similarly, looked at both augmentation and normalization, but they looked at it in combination. They looked at 4 different settings and performed a lot of different experiments, still in the histopathology section images though. And they kind of conclude that augmentation is always important. You could combine it with normalization. That might be that then the type of augmentation is not as important if you do a normalization in addition, but that you still need to do some augmentation. So to facilitate generalization, it is important to have varied training data, natural or artificially, or do data normalization. And in particular, this is important to facilitate generalization to external cohorts, because external cohorts would generally be more different than test subsets of the training data. So it might not be as important for generalization to test subset as it would be for generalization to external cohorts. And this robustness it entails is often critical if the system should be used in practice, because you won't have new data coming from the development data. You would have new data coming from new patients or new subjects or new settings. So it's critical with this robustness. The particular amount of data distortion or a normalization method should be adapted to the input type. Where I have exemplified it using histopathology images, but it might look a bit different, and the type of approaches you would use might be different for different input types. It might also be related to the prediction task. There will be a trade-off between facilitating the learning of relations that generalize well beyond the development data, so what we have looked at now, and on the other hand, not including relevant information for a prediction task. For instance, if color was important for a particular prediction task, it might decrease the performance due to extensive color distortion. But if it varies between the data set, it might still be positive. So it needs to be adapted. And it should be noted that this tuning subset, which is often called validation subsets in the machine learning community, they might not give the same trend as external data sets. For instance, in the experiment I just presented, we also tested the performance on tuning and test subset, and there it didn't seem that you should increase the amount of color distortion as much as 4 times. 2 times was the best there. But on external cohorts, it was beneficial to do more. And then coming back to the initial subject of evaluation, should decide everything on forehand, and when all is decided, then it's time to validate, and then we advocate for doing it, pre-specifying the analysis, the model, before doing the evaluation. I just want to acknowledge my co-authors of these 2 papers. There are also many others than this, particularly non-scientific people in the lab, which has contributed, but this is the co-authors. So thanks to them.
Thank you very much, Andreas, for an excellent and very, very interesting talk. And I'm sure many of the audience are aware that the Lancet and Nature reviews cancer. They are very high impact journals. Most AI researchers never get to publish in this journal. So congratulations with that, Andreas. We have several questions, but first I would also like to point out that we actually in NORA also had the podcast with Håvard Danielsen. So if you're more interested in these 2 papers and this work, you could also go into the NORA web pages over our podcast and listen to the podcast with Håvard Danielsen. Yes, I will read up a couple of questions. I also have some questions myself, but I will try to stick to the audience. I will first go to these questions about the requirements, the software and hardware resources. This is a question by Sabri, the software and hardware resources used in these sort of studies. Which infrastructure you had used in your study? You have local infrastructure at the DoMore project, right? How demanding is this?
Yeah, it is quite demanding. It depends, of course, on the network you use. So we have local infrastructure with, I think it's about 70 GPU cards now. For the final Lancet training, we applied this, or we have this set of 5 machines where we each have 8 GPU cards, where we train this model so that we kind of into one time on each solution, and across all 5 we get our final system. So yeah, we have in our experience, it has been nice to have access to the hardware ourselves. It is of course demanding, and many experiments has been undertaken, and yeah, that's.
And the software you typically used for these projects?
The software,
Yeah,
it's TensorFlow.
TensorFlow. Yeah, maybe we can follow this up. I mean, if you in Norway we have also national infrastructure. What would make you kind of choose the national infrastructure? Would that even be a possibility for you, or could you think of kind of situations where national infrastructure is more relevant or should be used?
Yeah, it could be relevant if we don't have the capacity ourselves. We have recently bought a DGX A100 to expand on the possibilities, particularly related to that. You saw that these images were quite large, so we tiled them up, and that might lose information. So we want to test out what if we could input larger tiles at higher resolution. For instance, we could also, there are of course details in this approach as well, and we could be interesting to use more tiles per patient when doing the training. That would also require more memory, GPU memory. Of course, if the national or international infrastructure would allow us to do much more, that would have been interesting. But since we have recently bought some high-end equipment for this, it might not be just yet.
Thank you. I'm sure Sabri will take notes. I will go on with the question from Vladimir. I assume the external data should be notably different from the development data. Is there a formal metrics, a distance measure, to estimate and assess such differences?
Yeah, that's a good question. I think that the main thing here is that we should think about when it's applied in practice. It would probably be applied at different places, and whatever is representative of where it will be applied, which has nothing to do with the development, that could be a nice external data set. So as long as there is no overlap at all, ideally not even between the, at least in the medical images, they're usually acquired at the particular hospitals or institutions. So there shouldn't be any overlap in the institutions, in the catchment area of the patients. And then you could have many good external cohorts, and I wouldn't say that they have to be that different in terms of any metric, but they should be kind of been objectively no overlap on a kind of large scale, so that geographically different would be a good way to start.
Exactly. One more from Vladimir. Does the survivability prediction take into account chemo and radiotherapy, or is it based on images only?
It's based on images only. So that's the kind of the main application of this model would be to determine who should receive adjuvant chemotherapy and who should not. In the validation cohort, it was a clinical trial, not a trial on a deep learning system, but a trial on a drug, which showed to have no effect. And then the nice thing about that is that the treatment is consistent. So every patient in the validation cohort received single agent chemotherapy, so a simple chemotherapy variant, and that means that we would not be it would not be a confounder in the validation then.
Exactly. Next question is from Jon Becker. You proposed a protocol for designing a deep learning study. Thinking of the even bigger picture, how do you propose we evaluate whether one protocol is better than the other as we move towards what is historically called a gold standard protocol in medicine?
Yeah, so this type of protocol is supposed to be the type of protocol we are talking about here is a study protocol or a validation study protocol. So it's particular to a particular system, a particular analysis of that system on a particular external data set. So the idea behind it is not the kind of it's not to sketch out the best method. It's to clearly state on forehand what is to be done and precisely how it is done. So in the clinical trials, you have similar protocols. The main thing there is, of course, that they do enrollment over many years and things can change and stuff. But here you should be able to define everything on forehand. So it's I guess it's the evaluation, the validation itself, which should then be used to measure which systems are the best, and then you should probably compare the systems as well. But that's not the aim of the protocol. It's just to define precisely what to evaluate on an external data set.
Exactly. I will read a question from Saba Milvaganam. The data in your study are mainly images, or you have other factors on the patients involved in your DL algorithms. Your predictions were related to the development of the cancer or extent of the tumor.
It was no other parameters there. It was only the images. Of course, they had a particular type of cancer. They had either colon or rectal cancer, and they had no metastasis in the set we analyzed. But they were just the images. So the aim was kind of going directly from this type of standard pathology images to patient outcome, and then kind of in there it seems therefore this I show this multivariable analysis where we compare and adjust for clinical and pathological variables, and that's important because then this model is not intended to replace everything. It's intended to supplement the existing working parameters. But in the colon cancer, it's not too many.
Exactly. And one last question before it's soon approaching 3 o'clock now, but I will take a question here. In the case of simulated development training datasets, what do you think the external datasets should preserve and vary from the development datasets?
Once again, what?
Yes, it's about the simulated development datasets. But what do you think the external datasets should preserve and vary from the development datasets?
Yeah, I don't think it needs to vary. I think it needs or it needs to be no overlap between the development and the external data. And as I mentioned earlier, geographical separation is a good starting point for that. But it could certainly be the same type of imaging equipment and the same type of patients or other subjects. Then, of course, the validation is specific to that setting. So that's a restriction you kind of have assumed then. But it's important that it's a representative. You kind of have some idea of what it represents, the external data set, because your result will be unbiased for whatever that represents. So if it's some biases in the external data, then that might not be good and might provide also misleading results if you don't know kind of the setting it represents.
Exactly. It is 3 o'clock. Unfortunately, there are several more questions and also many greetings. They say I will send it over to you, Andreas, but in general, people
Thanks.
are very happy about your talk. So thank you very much. And again, please go into NORA webpage and listen to the podcast. You could also sign up for the newsletter from NORA to get informed about future events. We have webinars every Friday at 2 o'clock. So thank you for a great presentation, Andreas.
Thank you for listening, all of you.
Thank you, and have a good Friday to everyone.
Thank you.